Many thanks for joining. Unfortunately, we have only one talk today from Margaret Wanjiku from Kenya. So Margaret, I will kindly ask that you share your screen. Margaret holds a bachelor's degree in biochemistry from Pwani University in Kenya. She is now doing masters in bioinformatics in the same institution. She is with EAN Bits at ICIPE and also a member of ISCB. So, Margaret, the floor is yours and you can take as much time as you need. All right, thank you very much. You may start. Margaret, hello. Can you hear me?
Hello, sorry about that.
Yeah,
Hello,
all right.
can you
Yeah,
hear
go
me?
ahead. Yes.
Okay. Good afternoon, everyone. My name is Margaret, as you have heard, and this is the work I am doing for my MSc project under the supervision of Dr. John Baker of Baker Hill Rehab and Dr. Wilton Binder of Pwani University. I can't move to the next slide. Okay, okay, there it is. Sorry. So I have been studying Magnaporthe oryzae, the causative agent of rice blast disease. It is rated as the most destructive disease in rice globally. It has been found to be highly genetically diverse. So I sought to understand the phylogenetic evolution and population structure of this pathogen in Africa. And this is because previous phylogenetic studies either included just a few isolates from Africa or only zeroed in on a few areas in Africa, for instance, East Africa as a region or a single country like Uganda. Therefore, my main objective was to infer the possible points of introduction and routes of migration of the pathogen in Africa. Could have been one or more, and that's why it's in plural. Um, my project was in two parts, but both parts started from here with 49 isolates. Amongst the 49, 43 had been collected from various African countries, and the remaining 6 isolates had been collected in Asia. In Asia, we had 3, and South America, we had 3, so making a total of 6. And of note, the 6 non-African isolates were added to enhance comparability within the data set. So starting from here to the first part of the project, going this way, the raw sequence reads were prepared for analysis by running them through FastQC to counter-check their quality before proceeding with downstream analysis. Once I ascertained the quality, I proceeded with downstream analysis. The reads were then mapped to a reference genome of Magnaporthe oryzae, MG8. After that, I performed variant calling and managed to get a total of 405, 383 genome-wide SNPs, which were used in the proceeding analysis. Here for the phylogenetic analysis and population structure analysis. So the population structure analysis included PCA analysis and admixture analysis. To the second part of the project, the main goal was to determine the distribution patterns of 178 effector genes obtained from a study published earlier this year by Latour et al. So the 178 genes were concatenated to form a reference upon which the 49 isolates were mapped. So again, we start here, moving this way to the second part. So they were mapped they were mapped to this reference. The 49 isolates were mapped to the reference, and then I proceeded to calculate the average coverage of each gene, of each of the 178 gene in each of the 49 isolates. I'm so sorry. So a gene was considered to be present in an isolate if 80% of its sequence was recovered after mapping. The results of this were then used to create a presence absence matrix, where ones represented presence and zeros represented absence. This matrix was then used to create a heat map so that we can compare distribution patterns between African and non-African isolates and make inferences from that. And so on to my results. I first wanted to look at the genomic locations of SNPs. And so in this particular density plot represented here, the peaks represent the genomic locations. You can see that we have peaks. First of all, the genome of M. oryzae has 7 chromosomes and a supercontig. So we wanted to see how the SNPs are distributed across all the chromosomes. And so the peaks here represent locations with the highest number of SNPs. And as you can see, supercontig 8.8 appears to have the highest number of SNPs, but upon further probing, it in fact has the least number of SNPs mapped to it and is the shortest of the chromosomes. What we did find, however, is that chromosome 1 has the highest number of SNPs, which is about 68,038. The rest of the chromosomes have between 36,000 and 66,000 SNPs, so that was a good distribution, and so we proceeded. To the phylogenetic analysis, this particular phylogram, you see that isolates from the same region cluster together, and East African isolates are represented in blue, West African isolates are represented in red, and non-African isolates are represented in green. Particularly, these 3 isolates appear to be significantly different from the rest, and we shall get into that as we proceed. Generally, you see that isolates from the same region, this is West African, for instance, in this particular clade here, cluster at the most, as opposed to the East African isolates. Whereas in this clade here, we have more East African isolates compared to the West African isolates. So for the PCA analysis, we started the initial, we did 2, and I shall explain why. So initially, PC1, this is the first PCA analysis. PC1 had a variation of 66.4%, while PC2 had a variation of 7.8% of the variation, making a total of 74.2 variation. However, okay, so these 3 isolates were the ones that are the green non-African isolates from the phylogenetic tree. So they were skewing the whole PCA plot. So from this is a bar plot of contributions of individual isolates to PC1 and PC2. So the red dotted line represents a reference line that corresponds to the expected value if the contributions were uniform. And from here, you see the isolates that I talked about have the highest influence on variability, while it is an isolate from Guyana, has the lowest influence on the variability. So we decided to remove the 3 so that we can see if the results will be different. And that's what we did. So this is 46 isolates, and this is PC1 and PC2. PC1 has represents a variation of 24.11%, while PC2 represents a variation of 11.78%. Now, the problem with this one is that the total variation explained is quite low. That is 35.89%. That is the problem. But however, we see clearly that the data set is divided into 3 clades very, very clearly, as opposed to the other one. And so this one just shows all the 46 isolates on the PCA plot, and you see that the ones from the Philippines, that remained So we removed the three that we removed were non-African isolates, and we remained with three. This one from the Philippines, these two from the Philippines, and this one from Guyana. But again, consistently, we see that isolates from the same region predominantly cluster together, as you can see here. And we see we have three clades. After that, I proceeded to do population structure analysis, and it was in two parts. The first one was admixture analysis, and the second one was the PCA analysis that I've just finished talking about. So for the admixture analysis, it would help in inferring an isolate's geographical origin based on the analysis of their genetic ancestry. So we ran 10 clusters of K, 10 iterations for each value of K that we ran. And so after that, we plotted the cross entropy criterion. Here we see that for the 10 runs of K, and this one will help in evaluating the quality of fit of the statistical model to the data using a cross-validation technique. So it will help us. The cross-entropy criterion ideally is supposed to help in choosing the number of ancestral populations, K, that best explain the data. And so here, what normally happens is that you choose the number. It's like a knee. You choose the number just before the value starts to increase. But this pattern, as you can see here, is not very clear. But we looked at K is equals to 5 because it appears to be the least before the value starts to increase. And so this is a bar plot of the admixture proportions where K is equals to 5. We have 5 clades. This one, the biggest one, which is represented by green. This one represented by violet. This one represented by yellow, blue, and red. And we see there are admixtures happening. And I think I will start looking at the ones that had little to no admixture, particularly these ones, the three here, the ones from Benin. There was also another one from these two from Togo. These three, the ones that we removed from the PCA analysis, they are considerably different from the rest of the isolates. And then we have this one from Burundi, and this one from Togo as well, and this from Nigeria, and there were two others from Nigeria as well, these two here. However, we see there are certain isolates like this particular one here has mixtures from all the clades. It has it's from Tanzania. There was also another one from Tanzania, this 32 that has a mixture of all of them. We also have another from Tanzania here that has a mixture. So generally we see isolates from East Africa displaying more admixture compared to the West African isolates. After that, so this is the second part of the project where I was looking at effector genes, and they secrete effector proteins. It's just a bit of for you to understand what effector genes are. Effector genes secrete effector proteins which are involved in a virulence capabilities of the pathogen towards the host. So a virulent effector genes will secrete proteins that will activate the host's immune response against the invading pathogen. So these are beneficial to the plant, to the rice plant, the host in this case. And the virulent effector genes secrete proteins that disrupt the cell signaling processes involved in immune responses of the host. So this, ideally, they are not beneficial to the host. So we wanted to see the distribution patterns of the effector genes in our isolate. So here we have the genes, and this the column, these are the isolates. So particularly the ones that were quite interesting were these two, this over here and this, which appeared to be present in all of the isolates apart from one, which was the isolate from Guyana, Guy 56. And something else also, when you look at the clustering of the isolates with the least number of genes, which appears to be this particular area, you see that the same genes that that are present in all the isolates are still present in the isolates with the least number of effector genes. And you see that they rarely have any other effector genes. So that could mean that these genes are being preserved, probably because they help the host to fight off the rice blast disease. Sorry. And so we can conclude that the genetic diversity does indeed exist among African isolates, and East Africa we found to be more genetically diverse compared to West Africa. And the pathogen could have been introduced to Africa through East Africa, and the pathogen has been evolving and continues to evolve. My recommendations would be to include more isolates from the northern and southern parts of Africa to get a better, clearer picture of the diversity within Africa as a whole, as opposed to looking at just east and west of Africa. And another one will be to explore the phylogeography of the pathogen in the globe, having included high numbers of African isolates, since I said in the beginning that global phylogenetic studies would only include a few, very, very few African isolates. So it's better to get a picture of the diversity of this pathogen in Africa so that we can get better suited solutions to fight the rice blast disease in Africa. I would like to acknowledge my supervisors, Dr. John Baker and Dr. Wilton Binder, and Dr. Geoffrey Onaga, whose data I used for this project. And I would also like to acknowledge Ian Beach, Isipe Pwani, Ajil, Ilri, Becker, and Fogarty and Erie. Thank you. Hello. Hello.
Albert, are you there?
Yeah, hello. Can you hear me?
We can hear you now, yes?
Yeah, sure. Margaret, I was
Yes.
asking that in summary,
Yeah.
can you tell us your main findings?
So generally, I found that East African isolates are more genetically diverse compared to the West African isolates, and so the point of introduction of the pathogen, or rather the rice blast disease, to Africa could have been East Africa.
All right.
Yeah.
Thank you very much. Margaret, there's a question from Frederick, and he would like to know the region or the chromosome, the violence
I can't hear
pathogen
you.
is associated with.
Hello.
There's a question in the chat box.
Okay.
Which region or chromosome is associated with violence in this pathogen?
Okay, so we were not focusing on virulence. That is not what we were trying to study in this particular study. Yeah.
Frederick, are you okay with it, or you like to expound on it? All right. So we still have some time left. You can ask your questions. Okay. Okay. So
Okay,
we think.
I have a question.
Oh,
So
sure.
why? So if the East African isolates are more diverse than the West African isolates, why do you think the West African isolates are not diversifying
The West African isolates are not diversified. Okay, so from
as
the
much?
yeah. Okay, so from the body of knowledge that exists, the research that has been done before showed that the pathogen, in fact, actually it was confirmed that it originated from Southeast Asia. And from the same study, it shows that as the pathogen, okay, at the source where the pathogen is originating from, there appears to be a lot more genetic diversity, and this is because it reproduces sexually. But as the pathogen continues to spread, it starts producing asexually, and so the lineages that result from from where the source is, where the pathogen was genetically diverse, they are termed as clonal lineages. Clonal meaning they are less genetically diverse, ideally.
Oh, okay, I see.
Yeah.
Thanks.
All right. Is there any more questions? Okay, then I would like to bring the meeting to a close. Verena, is there any announcements or anything you would like to say?
No, I think that I'd just like to remind the upcoming speakers to just please make sure that you submit your abstracts and your biographies on time so that we can advertise well ahead of the session, and then we are sure participation will be a lot better for your session as well. So please do that. And please try to not delay so that nobody else is delayed as well. And then, yeah, thank you, Margaret, for taking the time to do this talk. It was a really lovely talk. I look forward to hearing the next couple of talks. Thanks, everyone.
All right. All right. Thank you very much. Goodbye and see you in 2 weeks. Bye.
Goodbye.